This video is Chapter 9 Notes, Part 1. Chapter 9 is all about genetics, specifically Mendelian genetics. In terms of biology, I feel like there's one scientist everybody has heard of, which is Charles Darwin, and probably the second most famous biologist is this guy, Gregor Mendel. And Gregor Mendel studied genetics in the 1800s. And it's not important that we necessarily memorize the date, but more so think about the fact that in the 1800s, people had no idea what DNA was or how DNA worked. So Mendel was kind of a pioneer in that he figured that there was something moving from parents to offspring and that could affect how the offspring looked. Mendel worked in a monastery. He was kind of like the male version of a nun, and he did all of his research in the garden there. And just like Darwin, he collected a ton of data. And that's why he became to be very respected, because he had so much evidence to back up what he was saying. He studied different traits in pea plants, so you'll see a lot of genetics questions use pea plants as examples, because that's what Mendel was studying. So there's a bunch of vocab words for you in this unit, so to refresh your memory or maybe show you them for the first time. First one is genotype. When I talk about genotype, I'm talking about the genes. I'm talking about the DNA that's in an organism. So if I were to look at, let's say, this particular flower from the Punnett square, I would say that its genotype is homozygous recessive, meaning little r. To remind you, homo means the same, meaning big R, or little r, and hetero means different, meaning big R, little r. When I talk about the phenotype, I would be talking about what I physically see, the trait, and so again, using that same flower, its phenotype would be yellow in this case. I can also talk about a ratio. A genotypic ratio is how many of each do I have. So how many are homozygous dominant, how many are heterozygous, and how many are homozygous recessive. The order typically does look like that. However, in AP questions, you would not see both this option and 1 to 1 to 2. So they don't really try to confuse you or trip you up. They might put 1 to 1 to 0 as an option, which of course is different, so that would be wrong. But the ratio is how many of each are there. So I've got 1 big R, I've got 2 big R, and I've got 1 little r. So that would be the genotypic ratio. The phenotypic ratio is how many of each trait do I see. I see 3 that are pink, and I see 1 that is yellow. So my phenotypic ratio would be 3 to 1. You'll see a lot of questions asking about ratios. So now we'll do a little bit of practice. I'd recommend maybe you pause every so often and try to do them on your own. As you know, you have been doing Punnett squares since middle school, so I think it is a really bad assumption to assume that you already know how to do all of these questions, because in true AP fashion, some of them will get quite hard quite fast. So please do not be that person who assumes you know Punnett squares and just tries to envision them in your head. Always draw them, because what seems simple can quickly become much more complicated. I have done literally thousands, or maybe even more than that, Punnett squares. I still always draw them. It takes me less than 10 seconds to draw a Punnett square, and it pretty much always guarantees that I'll get the answer correct. So I would recommend that you don't try to just imagine them. Use scratch paper, use a blank piece in Notability, find something where you can do some practice. All right, here we go. In clownfish, having 3 stripes is dominant to 2 stripes. Marlin is heterozygous, so I know he's going to look like that. Coral is homozygous recessive. I'm going to quickly draw the Punnett square because I don't ever envision it in my head. That's a bad habit to get into. And once my square is drawn, I could easily answer 10 questions about that square. Here I've got 2. What fraction will have 3 stripes? 3 stripes is big T, so I've got 2 out of 4 of them will have 3 stripes. I can simplify that fraction to a half. Generally, the fractions are simplified. Question 2, what is the phenotypic ratio? Well, in my Punnett square, I've got 2 that have 3 stripes, and I have 2 that have 2 stripes, so my ratio is 2 to 2. 2 to 2 can be simplified because 2 can be divided by 2, both this 2 and that 2 can be divided by the same number, so the simplified ratio is 1 to 1. Next set. In bulldogs, having 8 whiskers is dominant to 6. 2 are mated, each of which has 6. Well, I know that 6 is recessive, so I am going to draw that Punnett square. I'll just pick any letter. And I can see in my Punnett square that all of the offspring will appear the same. So question 3, what fraction will have 6 whiskers? That is 4 out of 4. I can divide both 4s by 4, so I can simplify that to 1 out of 1. I can simplify that further to 1. Answer is 1. Question 4, what is the genotypic ratio? Well, I'm thinking about how many are big R, big R, how many are big R, little r, and how many are little r, little r. Well, I've got 0 of these, 0 of those, and I have 4 little r, little r's. That can be simplified by dividing all of those values by 4. So my simplified answer is 0 to 0 to 1. Now to introduce some new vocab. A carrier is somebody who is heterozygous, meaning they have the allele for a particular trait, but they are not showing it. So we say that they carry the recessive allele. They don't show that in their phenotype, but they are carrying it in their DNA. Something that is autosomal is not related to sex. What that means is that males and females have an equal chance of inheriting that trait. For now, the problems that you'll see are all autosomal, but that will change after you watch the following video. So in an autosomal trait, the trait will show up equally in male and female. It does not depend on the sex of the organism. A test cross is a cross done when you don't know the genotype of a particular parent. In this example, we have flowers and big P, I'm looking here, big P is dominant. So when I see a flower, in this case that's purple, with big P representing purple, I do not know when I look at that flower if that flower is big P, or if it is big P, little p. I don't know. Could be either. If I see a white flower, however, I do for sure know that its genotype must be homozygous recessive because it cannot have that big P. If it had that big P, it would automatically look purple. So in a test cross, we are always breeding to a plant or to an organism that is homozygous recessive. From the resulting offspring, I can then make a conclusion about the parent. So shown here are 2 possible outcomes from breeding my unknown purple plant with my white plant. If I end up with any offspring that are white, I know that my unknown parent had to be heterozygous, because that's the only way I could possibly get any white offspring. If all of my offspring are dominant, then I can conclude that my unknown parent must have been homozygous dominant. So a test cross is done when you have a dominant phenotype for which you do not know the genotype. More vocab for you. A recombinant is an offspring that looks different or has different genes than the parent. To recombine means that things are mixed together in a new fashion. So a recombinant offspring has DNA that is not identical to either parent. So shown in this cat cross, all of the offspring are recombinants. These 4 offspring are actually all the same compared to each other, but none of them are identical to a parent. My offspring, they are heterozygous for S and heterozygous for B. My parents, neither one is heterozygous for S, and neither one is heterozygous for B. So I would call all of these offspring recombinants. They are all different than the parents. True breeding is in some ways the opposite. A true breeding organism will only produce offspring that's identical to the parent. If I say that an organism is true breeding, that means that they are homozygous. They could be homozygous dominant, or they could be homozygous recessive. But if they are true breeding, they have only one possible allele to pass on. Like this one has to pass on a big A, and this one has to pass on a little a. So a true breeding organism has only one piece of DNA it could pass on, and thus it is homozygous. Some additional practice questions. Again, I would be pausing the video and practicing on your own. It's one thing to watch me do crosses, it's another thing to try them on your own. As I said, things will escalate quickly, so please practice even what seems simple, because it might not be as obvious as you think. In lions, having long whiskers is dominant to short, black feet is dominant to brown. Here I've got a male, he's heterozygous for both, and I've got a female who's homozygous recessive for both. I am looking for offspring that are recombinants. So I am looking for offspring that are different than the parents. All right, so I see that this is the same as a parent. This is the same as a parent. So I'm going to eliminate A and C because those are not recombinants. I can see this guy also is the same, which leads me to conclude that B must be the answer. I'm going to double check. I've got a hetero and a homo, doesn't match my parents. I've got a homo and a hetero, does not match my parents. Therefore, the answer is B. Both of those offspring are recombinants. In border collies, black coat is dominant to red. A breeder has a black male. The breeder wants to use the dog, probably make some good money, and so she breeds him in a test cross. All right, so I automatically know the definition of a test cross means I have an unknown parent. I am going to try to figure out, is that parent hetero or homozygous? Always in a test cross, I am breeding to a second parent that is homozygous recessive. That is the definition of a test cross. Question 6 is asking me, what is the expected ratio if the male is purebred? All right, well, if he's purebred, the definition of purebred is homozygous, so that would mean that these two are both big B, little b. So if the offspring was purebred, what is my expected phenotypic ratio? How many blacks would I have to how many reds? Well, all 4 of them would be black, and I would have 0 that are red. I can simplify that by dividing both of those by 4, which would give me a 1-to-0 ratio. Question 7. After breeding, one of the puppies has a red coat. Okay, so somehow I ended up with offspring that are little b, little b. So the only way that I could have gotten those little b's was if my parent here was big B, little b. That is the only way to get offspring that are little b, little b. So question 7 is asking me, is the dog purebred? Well, if it's big B, little b, that is heterozygous, so no, it would not be purebred. A little bit about generations. We sometimes see these described in questions where the P generation just refers to the parent generation. The F1 is the first set of offspring, so I would cross 2 parents to get offspring. Sometimes you'll see then that all the offspring are identical, and so a question then asks you, well, what if those offspring bred with each other? And we would call that resulting generation F2. So just a little bit of lingo that you might see in some of the questions. It's just talking about P, F1 and F2 are just talking about different generations. Moving on, we'll end this section with pedigrees. A pedigree is a chart that's used to track a trait through a family. In the pedigrees, squares represent males, circles represent females, and everybody shaded has a particular trait. So I can start to identify relationships between the people. Here I have a male, here I have a female. That male and female mated, which is my horizontal line connecting them, and they had 4 children. Of those 4 children, I can see that 3 of them are male and 1 of them is female. The shaded individuals have the trait, so dad had the trait, daughter has the trait, and this particular son also has the trait. When reading a pedigree, I can determine if a trait is dominant or if a trait is recessive. To remind you, this word autosomal means that a trait is not related to sex, and therefore it gets passed down equally between males and females. This particular slide identifies how to help you figure out if a trait is recessive or if a trait is dominant. If a trait is recessive, then I might have entire generations where I do not see the trait. So males and females are equally affected. That's the definition of autosomal. If I see entire generations without the trait, then I know that it's a recessive trait. Affected kids don't have to have an affected parent. So here is a kid who is affected, but neither of his parents are affected. On the contrary, in a dominant pedigree, every affected kid has an affected parent. It does not skip generations. So you'll see questions that ask you, is the trait dominant or recessive? Best way to tell, are there entire generations that don't have the trait? If so, it's recessive. A few pedigree practice. Question number 1, who is 5's mother? Again, I would pause it and try these on your own first. 5's mom is a circle, so therefore it's 3. Is this pedigree for dominant or recessive? Here I have an entire generation where nobody has the trait, and then poof, the trait appears in number 5. That tells me it must be recessive. I'm now asked to figure out the genotype of each person. Well, I know that this is a recessive pedigree, so therefore my shaded individual must be little a, little a. Everybody who is not shaded must have at least one capital letter. If they had both lowercase, they would be shaded. So they have to have at least one capital. I also know that individual number 5 got a little a from mom and got a little a from dad. That's the only way to be little a, little a. So mom has to be heterozygous. Dad also has to be heterozygous. For kid number 6, I'm a little bit unclear. If I draw the Punnett square from the mom and the dad, I can get my shaded kid. There she is. And I can also get homozygous dominant, or I could get heterozygous. So it is unclear to me exactly what this kid is. They could be homozygous, or they could be heterozygous. I actually don't know. So in that case, I would say it could be either homozygous dominant or heterozygous. Definitely they're not recessive because they're not shaded. But of those two, it could be either. Question number 7, if 5 has a baby, what's the chance of passing on the allele? Well, she's going to pass on one of the alleles. So she could pass on little a, or she could pass on little a. That's the only option she has because she's little a, little a. So she has to pass on a little a, little a. So the chance of her passing on the allele is 100%, or I could say 1, meaning 1 out of 1. That's the only option she has. Little bit more practice. Number 8, the genotype of 2. Well, I'm just going to work through this entire pedigree to try to figure out what is everybody, and then I'm going to go back and answer the questions. This trait is dominant. I know every single shaded person has to have a dominant allele. I know every unshaded person has to be homozygous recessive, because if they had a dominant, they would be shaded. I'm now going to work my way up or down, it doesn't really matter, to try to fill in all of my missing pieces. I'm going to start here with individual number 7. Individual number 7 has a dad who only has little a, little a. Therefore, dad must give a little a. So I know individual 7 has to be big A, little a. Looking at individual 5, I'm also not quite sure until I look up and I see that number 5's mom is little a, little a. Number 5's mom had to give a little a. So number 5 must be heterozygous. And then individual number 1, I can see that they had some kids, and those kids got 2 little a's. So they had to get a little a from mom. Yep, that's all she has. And they had to get a little a from dad. So this dad here has to be heterozygous. So to conclude, I'll answer the questions. Number 8, the genotype of 2 is little a, little a. The genotype of 1, big A, little a. I find it easier to label my pedigree and then go answer the questions rather than trying to figure out things simultaneously. Number 10, who is 5's sister? The females are circles, so therefore the answer is 4. And then if person 7 marries a woman who is heterozygous, so they're going to get married, what fraction are expected to have this trait? I don't imagine things in my head. I draw them. It takes me a few seconds, but is much more likely I'll get the answer correct. This particular trait is dominant, so all of these 3 kids will have that trait. So 3 out of 4 kids will have that trait.